Good morning, everyone. This is the second video on evolution. In the first part, we saw how we could build phylogenetic trees. To do so, we looked at different species and we compared different characteristics. And the basic principle was the following: if two species have more characteristics in common, then they are more closely related. In this part, we're going to do something similar. We will be building phylogenetic trees. The only difference is that instead of looking at characteristics such as bones, anatomy, we're going to have a look at molecular characteristics. And for example, that's the one we will start off with. We're going to look at DNA sequences. That is why part number three, use of DNA sequencing to build phylogenetic trees. And the first thing we're going to look at is DNA sequencing methods. To understand DNA sequencing, we need to do a few recaps on how DNA replication works. So we're going to have a look at this diagram that we saw in the beginning of the year. So this is how replication occurs with DNA polymerase. Two things I'd like to point out. The first one is that on the sugar here, the deoxyribose, there is an OH group. This is very important because if this OH group was not present, then replication would not be possible. You remember that phosphate groups, two phosphate groups will leave the entering nucleoside, and the breaking of this bond here is going to release energy, and that will enable this phosphate group to bind to the OH group. And at the end, you have the bind, which is here, and you see that thymine binds with adenine, for example. So what's really important to remember is that the OH group is needed for the entering nucleotide to bind to the previous one. This OH group is needed. And just for the reference, it was from the Campbell book, this diagram. The DNA sequencing technique we're going to have a look at is called the Sanger sequencing method. It's based on the selective incorporation of chain terminating D-deoxynucleotides by DNA polymerase during in vitro DNA replication. Okay, this sentence probably sounds very barbaric, so we're going to try and break it down, and at the end of this video I can assure you that you will understand what this means. So the first thing we're going to look at, so the technique is called the Sanger technique, and we're going to look at this first word, D-deoxynucleotides or dideoxynucleotides. So what is that, you may ask? Well, it looks like this. In the previous recap, we said that nucleotides have a OH group. So this is the classic nucleotide, a deoxynucleotide. Well, in Sanger sequencing, scientists were able to create nucleotides where instead of having a OH group, it's only a hydrogen. So you can see that there's a difference. In D-deoxynucleotide, there is no oxygen. So instead of having the hydroxyl group, it's simply a hydrogen. And that's going to make a huge difference because, as we saw earlier, for the new nucleotide to bind to the previous one, you need this OH group. So this means that this is going to alter replication. It's going to stop replication. And we're going to see how in more detail with some diagrams. To explain how the Sanger method of DNA sequencing works, I'm going to use a YouTube video which has this exact title, the Sanger method of DNA sequencing. You have the link in your booklet, so you can watch the whole video if you wish. That will probably help you understand. I'm just going to focus on a few key points which I think are the most important ones, but obviously it will help you if you watch the whole video. The first step to this technique is amplification. Let's say that this is the DNA fragment you want to sequence. Well, the first step, you need to do thousands of copies. How is this done? By using PCR, polymerase chain reaction. We studied this in the beginning of the year, so you can see behind it, the video illustrates that these are other copies of this fragment that scientists want to sequence. Once the fragments have been amplified, the second step is to heat up the system, and that will separate the two strands. We can see that the two strands have been separated, and that is thanks to heat. Next step, just like in normal replication, a primer is added so that replication can take place. Once the primers have been added, these strands with the primer will be dispersed into 4 tubes. In each of these tubes, there will be the same amount of this type of strand of DNA plus the primer. There will be the same amount in tube 1, 2, 3, and 4. Same amount of this segment with the primer. Afterwards, DNA polymerase is added in each tube. DNA polymerase, as you know, will perform replication. So again, same amount in each tube, and in this case, DNA polymerase has been added. The DNA polymerase will be followed by, it's written here, all 4 DNTPs. So these are the classic nucleotides, the ones with the OH groups. So they're really the nucleotides you've always been studying when we talked about PCR or classic DNA replication. These are the 4 nucleotides with the A, T, C, and G bases. And these ones, they have the OH group. They're the classic ones, the normal ones, if I may say so, with the OH group on the sugar. After the DNTPs are added, the DDNTPs, so that's what we just saw, this time it's the nucleotides without the OH group, so nucleotides which have only a hydrogen on the sugar. But instead of putting the 4 types in each tube, this is where it differs. In each tube, there's only going to be 1 type of these DDNTPs. So this is what is shown in the video. So each tube will have a different DNNTP. So this is shown by different colors. Each tube will have a different DDNTP. There we are. Now then, let's focus in what happens in one of the tubes. So let's focus on one of them. In one tube, we have these elements. DNA polymerase is going to add the nucleotides. So this is a very simplified diagram, but you imagine that we've already studied this. DNA polymerase will add the nucleotides one by one. And it's going to stop at this point here. Why did it stop? Because the DDNTP has been placed, and therefore no new nucleotides can be added. When DNA polymerase arrives to this part, it can't add a new one. It can't add a new nucleotide because here there is no OH group. The OH group, which should be here, is missing. Therefore, DNA polymerase simply stops here. In each tube, different fragments of different lengths will be produced, depending on where the DDNTP will be paired. Because if you have, for example, 3 A's in a sequence, well, the DDNTP with a thymine base can pair up with any of these A's, and it can stop at different moments. So in each tube, so in this tube there will be different sequences for a type of DDNP. Same thing here, same thing here, same thing there. And at the end, you will be able to, thanks to electrophoresis, you will be able to know the lengths of these different fragments which have been produced, and therefore you can deduce the sequencing. So let's have a look at how it unfolds. So you know that in electrophoresis a current goes through the agarose gel, and you can see that for the different types of nucleotides we've got different fragments. At the end you simply have to read from bottom to the top, and that will give you the order of the sequence. Okay, so as I said, you need to read from bottom to top, and that gives you the sequence. So the bottom one is A, then we have G, then we have C, then T, etc. And by doing this, at the end, by going up the electrophoresis with the different size of fragments, you will know which nucleotide is which, and you end up with the complementary sequence. That's important to say, that the sequence that we obtain in the end is the complementary one. Therefore, to deduce the second one, you simply have to do the Chargaff rule, so T, C, G, etc. Okay, in your booklet, I found another diagram which sums up this technique. To be noted that you can also analyze using a laser beam through the electrophoresis. That will also give you the nucleotides. So in this example, this is basically what happens in the 4 different tubes. And if you start with this sequence here, DDT will stop at the first A, it will stop at the 4th A, and it will stop at the 5, 6, 7, 8 at the 8th position. So with these fragments, we know that as the DDT corresponds to where an adenine is, we know that the first one is adenine, that's what we have here. The 4th one will also be adenine, that's what we have here. And the 1, 2, 3, 4, 5, 6, 7, 8 will also be adenine, that's what we have here. In the second one, it stopped at 2, so we know that DDA will be complementary with a T. So we know that the second will be a T, and we know that 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6 will also be a T, and etc. and etc. I really encourage you to go through this, reread, even maybe cut out some nucleotides for yourself and try and see how it works out. That might be a good way to memorize how this technique works. So there we are. That's what is called the Sanger sequencing technique. So you now know how it works. 3.2, building phylogenetic trees using DNA sequences. To do that, so you know now how the sequencing technique works. We're simply going to do an example to build a phylogenetic tree with these sequences. So the example we're going to look at is from Bac exercise 2016. So this example is the following. We're looking at the sequences of the same DNA segment, and it's for the gene of the beta casein in milk. So all these mammals produce milk, and they all have a similar gene. So let's have a look. We have the sequence for the sperm whale. So all these sequences have been determined with the Sanger technique, for example. So it goes A, G, T, C, 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 etc. How does this table work? Well, basically for the camel, when you've got lines like this means it's the same. So you need to imagine that here would be C, C, C. And when you've got letters, it's when you have a difference. So we simply have to count the differences. If we take the example between the sperm whale and the camel, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. These two sequences have 11 differences. 11 nucleotides are different. Just like for classic phylogenetic trees, we still have the same basic principle. In this case, the less differences there are between the same DNA sequences for two species suggests that they are more closely related. If the DNA sequences are more similar, then they are more closely related. In the previous examples, it was, for example, a bigger number of bone similarities. In this case, it's a bigger number of common nucleotides. But the principle is the same. Okay, the first question of this back exercise is the following. Copy and complete the table below showing the number of differences between the DNA sequences of these 7 species. Just like for other characteristics, you need to do a characteristic grid. But instead of counting number of bones, for example, in this case, it's simply the number of differences between the DNA sequences. Now then, to answer this first question, we've got the DNA sequence grid, and we've got this table. So you've got some squares which are empty, and you will have to compare. So for example, you need to compare the cow and the camel. So this means that you will have to compare these two lines. You need to be careful of something. The dots mean that it's the same nucleotide as the sperm whale. You always must remember that. So for example, the camel has a T, the cow will have an A here. So that is one difference. So I'm going to repeat that. The dots, they're telling you that it's the same nucleotide as the sperm whale, and that's true for all the species. So our first example here, if we compare the first nucleotide, the camel has a T and the cow has a dot, so it's going to be the same one as the sperm whale. So A and T, that is one difference. And you're going to have to do that for all the nucleotides. So I'm just going to give you the answers. So 14, 12, 8, 11, 11, 8, 3, and 10. So I'll let you check that with by doing this technique as I just showed you. We are now going to answer question number 2. So in this question, we need to determine the evolutionary relationships. So now that we've got the complete grid, we have all the information we need. So we know the differences between the DNA sequences. So the species that we're going to use as the main one, if I may say so, is the sperm whale. So we're just simply going to say, compared to the sperm whale, which species is closer? So we don't need the rest of the grid. We only need this first column where we've got the sperm whale and all the other species. So it's logic that there is a 0 here, because between a sperm whale and a sperm whale, they'll have exactly the same DNA sequence. We're going to build the branches. And now we need to compare and say which one is more closely related. You remember the basic principle is the following. If there are less differences, then they're more closely related. So these numbers here, they are the differences in terms of nucleotides for the sequences of all these species. So we need to simply find the smallest number, which in this case is 2. So the closest species to the sperm whale amongst this list of species is the porpoise. Okay, let's have a look at what it would look like on our phylogenetic tree. So we will have the sperm whale here. You have an elephant and a swimmer to see how big it is. So it's huge. So the closest species which is related among this list is the porpoise. Then we have to go in the column and see which is the next smallest number. So it's going to be 3. It's the hippopotamus. So we'll place the hippopotamus just next to the porpoise. Then we need to have a look at the next one. So 2, 3, seems to be 8, the next one. So it will then be the cow. The cow will be placed as such. Then we have, what's the next one? We have 8. Then we have 10 will be the pig. So the pig will be placed next to the cow.
I'm Peppa Pig.
Then the next one is 11, the camel. And the last one is the rhinoceros with 12. So we can see that we've established the relationships. The more you go towards the left, the less closely related they are, and that means that they share a common ancestor, but a much further in time ancestor. The porpoise and the sperm whale, they share a much closer common ancestor. So these common ancestors branches, they stay the same. It's the same principle as for the previous phylogenetic trees that we have studied. Last question. Explain why it is better to compare DNA sequences rather than amino acid sequences to establish the evolutionary relationships of different species. Well, it's quite simple to explain. You know that when we talked about DNA, which will be then used to make RNA and then RNA to proteins, you learned that the genetic code is redundant. That means that there are different DNA and RNA sequences which will code for the same amino acid. So you will be able to see more differences in the DNA sequence than in the amino acids. So this is what you can write. The few elements, that's from the marking scheme. So the genetic code is redundant. Some amino acids have more than one code. So some mutations produce no change in the amino acid sequence of proteins. And you could almost add, whereas these differences would be shown in the DNA sequence. That's all for DNA sequences and phylogenetic trees. I will see you next time.
Genes, phylogenetic trees, comparing HIV, V. We're talking about genes, phylogenetic trees, comparing HIV, V. How to analyze it? Nucleotide sequence alignment. First amplify a DNA region, then you compare the revolution.